Hi, hello students. Welcome to Mass Physics Seminar. This is your MRK Zoology faculty. So now we'll continue part 2 in female reproductive system. Okay. So previous class లో female reproductive system part 1 మనం డిస్కస్ చేసాము. సో హ్యూమన్ రిప్రొడక్టివ్ సిస్టంలో ఫోర్త్ క్లాస్ ఇది పార్ట్ 2. సో దెన్ కంటిన్యూషన్ ఆఫ్ ఫీమేల్ రిప్రొడక్టివ్ సిస్టమ్ ఇట్ ఈస్. కంటిన్యూషన్ ఫీమేల్ రిప్రొడక్టివ్ సిస్టమ్ కంటిన్యూషన్ చేస్తున్నాం ఇక్కడ. Okay, so in part 1 we are discussed about what is the total human female reproductive system and that system contain organs. So human female reproductive system లో ఉన్న organs ఏంటివి అంటే one pair of ovaries, one pair of oviducts, accessory glands, and what is uterus, next vagina and female external genitalia. We are clearly discussed about this. And pair of ovaries, we are discussed the clear information ovaries and ovary shape, color and internal structure, each ovary made by how many layers means we are clearly discussed that points. Okay, and next we are clearly discussed that oviducts, its length, and which parts are there in the fallopian tube, and we are clearly discussed that points. Next uterus parts, then the uterus wall is made by how many layers. Okay, which one is the broad, which one is the narrow portion, what is the shape of uterus, we are clearly discussed that thing. Next vagina lining and vaginal canal and birth canal formation. Birth canal is formed by vaginal canal and cervical canal. These two canals are involved in the formation of birth canal. Through the birth canal, the baby comes out from mother uterus to outside. Okay, so right, and after that next we'll discuss the condition, and up to last class we have discussed that vagina and female external genitalia, and after that we'll discuss female accessory glands. In the female accessory glands we'll discuss, okay, these are also called as a female reproductive glands. Female reproductive glands. Okay, so total 3 types of female reproductive glands are there. 3 types of glands. In male reproductive system, 3 types of male reproductive glands are there. Okay, and these 3 types of male reproductive glands are present and associate to the complete reproductive organs in male. But in female, these 3 types of in these 3 types of female reproductive glands, 2 types of female reproductive glands are associated with the female reproductive organs in the external genitalia, and 1 type of reproductive organs, reproductive glands are not associated with the directly female external genitalia. So these are, what are the 3 types? First I list out that 1 type of female reproductive glands. 1 type of glands are the Sken's glands. Sken's glands, and it is only one Sken's gland structure, that's enough. Next Bartholin glands. Bartholin glands, these are paired. Bartholin glands, these are paired. And third type of female reproductive glands, mammary glands. Third type of female reproductive glands, these are the mammary glands. These are the mammary glands. Okay, so these 3 types of female reproductive glands are there. Sken's glands are there, Bartholin glands are there, mammary glands are there. Actually, in this, some glands are homologous to female reproductive organs. Some female reproductive organs are homologous to male reproductive organs. So I list out that. Okay, for example, here homologous organs. Homologous organs. Okay, for example, here clitoris is there. Clitoris in female reproductive system, it is homologous to penis in male reproductive system. Next, what is Skens gland is there. Skens gland, it is homologous to Cowper gland or prostate gland. Cowper gland or prostate, and Bartholin glands in female reproductive system. Bartholin glands, these are homologous to bulbourethral glands. These are homologous to bulbourethral. These are homologous to bulbourethral glands. These are homologous to bulbourethral glands. These are okay. Clitoris is homologous to penis because structurally these are made by the same tissue. Okay, and functionally different, right? Skens glands are homologous to Cowper glands, and Bartholin glands are homologous to bulbourethral glands in male reproductive system. And here some what is in female reproductive system, labia majora. Labia majora, it is homologous to scrotal sacs. Scrotal sacs. Reason, with inside the scrotal sacs the testes are protected. Inside the labia majora, okay, inside the labia majora the total uvula or vulva, this is there, that is get protected. The total female external genitalia is protected inside the labia majora. So these are the some important glands here. Okay, these are homologous. Okay, right. And where the Skens gland, then come to the point, and location and function of this gland, Skens and Bartholin and mammary glands will discuss. Say Skens glands. The Skens glands are present, what is, anterior to the vaginal orifice. For example, just in the previous class we are discussed that external genitalia. It is the labia majora and labia minora, and it is the vaginal orifice, it is the urethral orifice. And anterior to the what is vaginal orifice, the Skens glands are there. Skens gland is present. Skens gland, it is present. And what is the function of this Skens gland? Means when it is stimulated during the copulation or during the mating or during the copulation or during the intercourse, it gets stimulated and which is secrete some secretions. That secretions are helps to lubrication of vagina. Okay, so it is the Skens gland. Next, second type of glands are there. These are the Bartholin glands. The Bartholin glands are located at the posterior part of the vaginal orifice. Okay, these are the Bartholin glands. These Bartholin glands are okay present at the posterior to the vaginal orifice, okay, and which are helps to lubricating the vaginal orifice always. Always the vaginal orifice is present in lubricating in nature, the presence of these Bartholin glands. These Bartholin glands, these are the functions of what is these Bartholin glands in the inner line. These are present inside the what is vaginal orifice. These are present inside the vaginal orifice. Okay, so these are the what is the Skens glands and the Bartholin glands function. Especially the Bartholin glands are more active than in primates, in non-primates. Reason, in non-primates the Bartholin gland secretions are giving some colors. They are showing some colors. That colors are okay. That colors are sorry. Along with that smells. Okay. So compare with colors. I'll show the best word, smells. So to that smells, that male non-primate attracting to the female non-primate. For example, cows or buffaloes, these are there. So when the female cows comes to the heat, okay, some the Bartholin glands are get activate and which are secrete some secretions. That secretions are the Bartholin gland secretions. That secretions are giving some smells. So to that smells, that male bulls are attracting to the female what is cows and buffaloes. Understood. But in human female, these Bartholin glands are not giving smells, and just it is helps to lubricating the vaginal orifice. Vaginal orifice it is. Vaginal canal is always lubricated by the what is secretions of Bartholin glands. Okay, so this is the what is this Skens glands and Bartholin gland glands function. Next, mammary gland. See, the mammary gland is the major female reproductive gland. It is mammary gland. It is the major mammary gland. Okay, it is the major female reproductive. It is the major female reproductive gland. It is okay. So one pair of mammary glands are there. One pair of mammary glands. Okay, one pair of mammary glands. These mammary glands are modified sweat glands. Okay, where the mammary glands are located? Location. These mammary glands are located at the breast region, at the thoracic, ventral to the thoracic. That portion is called the breast portion. In the breast portion region, that one pair of mammary glands are there. Actually, these are present at the breast region, breast portion, anterior, ventral to the thoracic region. And here the mammary glands are modified sweat glands. These are the modified, these are the modified sweat glands. These are the modified sweat glands. It is very important thing. And only two sweat glands are get modified into the mammary glands, the presence of some female reproductive hormones. Okay, which hormones are has major role in the development of mammary glands from the puberty age? Okay, estrogen and progesterone hormones are has major role. So I'll explain that hormonal regulation of development of mammary glands. Okay, hormonal regulation it is. Okay, I'll clearly explain this. Hormonal regulation. Hormonal regulation it is. Okay, generally the mammary glands development in a female, human female, it is started from the puberty age. ఓకే, అంటే మమ్మరీ గ్లాండ్స్ డెవలప్మెంట్ హ్యూమన్ ఫిమేల్లో ఎప్పుడు స్టార్ట్ అవుతుందంటే ప్యూబర్టీ ఏజ్లో స్టార్ట్ అవుతుంది. అండ్ వాట్ ఈస్ ద ప్యూబర్టీ ఏజ్ ఇన్ హ్యూమన్ beings, either in male or in female, means 12 to 14 years age is the what is puberty age. Okay, at from puberty age, from puberty, from puberty age, what is the puberty age means? 12 to 14 years age is the puberty age. In some human beings, the puberty age it is begins at 12th year or 13th year or 14th years. In rare condition, that is may delay, means in 15th year and 16th year also in human females the menstrual cycle will begin. In human females, menstrual cycle begin after 14 year also, in some very rare, but in some persons generally the menstrual cycle will begin from 12, in some females 12th year, in some females 13th, in some female human beings 14th year. This is the puberty age. Okay, it is development start from puberty. From the puberty age the mammary glands are started. So at puberty age, at the puberty age, hypothalamus is there. Okay, hypothalamus is there. Generally The hypothalamus can release gonadotrophin releasing hormone. These gonadotrophin releasing hormone stimulating to the anterior pituitary. It is stimulating to the anterior pituitary gland. Anterior pituitary gland. Now, this anterior pituitary gland can release gonadotrophins. These are released gonadotrophins. These are released gonadotrophins. These gonadotrophins are, one is the FSH, means follicular stimulating hormone, and the second one is the LH, luteinizing hormone. These two hormones are released. Okay, the follicular stimulating hormone, which is helps to development of what is Graafian follicle, means primary follicle, it is developed into Graafian follicle. The presence of Graafian follicle, Graafian follicle development is takes place by the what is follicular stimulating hormone, and it is also initiate the oogenesis. It is also initiate the oogenesis process. Okay, means oogenesis means production of ova. Next, luteinizing hormone, which is helps to, which is act on Graafian follicle and also helps to, first it is helps to ovulation, and which is maintain the empty Graafian follicle, that is the corpus luteum. Corpus luteum, and it is from the corpus luteum. Okay, from the corpus luteum that main hormones are released from the corpus luteum, estrogen and progesterone hormones. Progesterone, estrogen, progesterone hormones are released. Estrogen and progesterone hormones are released. Estrogen and progesterone hormones. Okay, I'll write once again this. Okay, right. So luteinizing hormone, which is what is causes to ovulation, it is first function, ovulation, and also maintain the corpus luteum. It is also maintain the corpus luteum. From the corpus luteum, okay, two hormones are released. That two hormones are, one is the estrogen hormone, estrogen hormone. Second one is the progesterone hormone, progesterone. See, the estrogen progesterone hormones are helps to development of mammary glands. It is development of mammary glands. Development of mammary gland. This is the hormonal regulation of mammary gland development. Okay, I'll give the entire information. See, what is the shape of mammary gland and what are the internal structures of mammary gland? Okay, we'll discuss. Actually, at the time of at the time of puberty age, at the time of puberty age, the mammary glands are actually in the thoracic region. What is the large fat bodies are there here, that fat bodies are get modified into the mammary glands actually. Okay, fat bodies, see, this is the mammary gland structure. Okay, the mammary gland, inside the mammary gland, what are the mammary tubules, mammary lobules are there. So it is total fat bodies. These are the total fat bodies. Okay, these are the fat bodies. It is total fat. Okay, after that, inside the what is mammary gland, mammary lobules are there. in each mammary gland 15 to 20 mammary lobules are there. Okay, 15 to 20 mammary lobules. It is the mammary lobule. 15 to 20 mammary lobules. 15 to 20 mammary lobules. These are 15 to 20 mammary lobules are there. These mammary lobules 15 to 20. It is clear point. Okay, like this. In each mammary gland, these 15 to 20 mammary glands are there, mammary lobules are there. See, in each mammary lobule, okay, in each mammary lobule, the fact modified structures, alveoli are there. These alveoli are look like a bunch of grapes. These alveoli are look like a bunch of grapes. These are look like a bunch of grapes. In each and every mammary lobule, these bunch of grape alveoli are there. These are the alveoli. It is the bunch of grapes. These are look like a bunch of grapes. Okay, it is look like a bunch of grape. Okay, bunch of grapes. It is look like a bunch of grapes, and these bunch of grapes are internally connected by, okay, internally connected by mammary tubules or mammary lobules. These are all are small mammary lobules are there. These all mammary lobes are fuse and form the one large mammary tubule. These are the mammary tubule. It is the mammary tubule, mammary tubule. Okay, okay, here, and it is also called as a mammary ductule. It is also called as a mammary ductules. Okay, the group of mammary ductules. Okay, the group of mammary ductules. These are fuse and form the one large duct. The large duct is called mammary duct. The large duct is called, that is the mammary duct. Okay, ductules. Small are the small diameter are ductules. The small ductules are again fuse and form the one large duct, that is the mammary duct. Say the mammary duct has, it is open into the one large wider portion. The large wider portion is the mammary ampulla. That is the mammary ampulla. It is, that is the mammary ampulla. Okay, see the mammary ampulla, it is again open into the one narrow duct, that is the lactiferous duct. That is the lactiferous. That is the lactiferous duct. Okay, the lactiferous duct, which is open out through one small opening. Through this opening, it is open out through the nipple. So the through which structure the lactiferous duct will open out, that is the nipple. That is the nipples. Okay, and surrounding of the nipple, the pigmented portion is there. Surrounding of the nipple, the pigmented portion is there. The pigmented portion is called here alveoli. The pigmented portion surrounding the nipple, this is the alveoli. Generally, it is not alveoli, it is also areolae. Generally, it is considered as a areolae or areoli, or sometimes it is also like areolae. This one. See, these areolae in some persons it is appeared in black color or bluish color. In some persons that surrounding the nipple, gray color also there. Okay, depend on the pigmentation within the body of that person. See, finally, okay, finally the milk is comes out from inside mammary glands to into the baby mouth by the sucking process. So this is the mammary gland. Once again I'll explain to you. Generally in our examination some questions are asked. At the puberty age in a human female, which hormones are helps to development of mammary glands? Okay, ఇక్కడ టూ హార్మోన్స్ యూజ్ అవుతాయి, ఈస్ట్రోజన్ అండ్ ప్రొజెస్టెరాన్. ఓకే, ఈస్ట్రోజన్ ప్రొజెస్టెరాన్ హార్మోన్స్ అనేవి మమ్మరీ గ్లాండ్ డెవలప్మెంట్కి యూజ్ అవుతాయి. ఫస్ట్ థింగ్. సో ఎలా మమ్మరీ గ్లాండ్ డెవలప్ అవుతాయి అంటే దట్ మోడిఫైడ్ ఫ్యాట్స్. ఇనిషియల్గా మమ్మరీ గ్లాండ్లో స్ట్రక్చరల్ అండ్ ఫంక్షనల్ యూనిట్ ఆఫ్ మమ్మరీ గ్లాండ్స్ అని వేటిని అంటారంటే ఆల్వియోలైస్ అని అంటారు. రీజన్ ఏంటి అంటే ఈ ఆల్వియోలైసే మిల్క్ని ప్రొడ్యూస్ చేస్తాయి. కాబట్టి స్ట్రక్చరల్ అండ్ ఫంక్షనల్ యూనిట్ ఆఫ్ mammary gland is the alveoli. These all alveoli are the modified fat bodies. That is clear. Okay, ఈ ఆల్ ఆల్వియోలైస్ మొత్తం కూడా మోడిఫైడ్ ఫ్యాట్సే. ఓకే, సో ఇది ఫస్ట్. ఈ ఆల్ దీస్ ఆల్ ఆల్వియోలైస్ ఆర్ లుక్ లైక్ ఏ బంచ్ ఆఫ్ గ్రేప్స్. గ్రేప్ బంచ్ లా కనిపిస్తుంది. సో అండ్ ఈచ్ అండ్ ఎవ్రీ alveoli it is attached with mammary ductules. Small ductules are there. The group of small ductules are fused and form the larger duct, that is the mammary duct. Again this mammary duct is open into the one swollen structure. The swollen structure is the mammary ampulla. Within the swollen structure of mammary duct is ampulla. Within the ampulla very less time or some time the milk can stored here. The milk can stored in the ampulla, and after that by the contraction and relaxation of this wall of this mammary duct, this milk can eject through the lactiferous duct into the infant baby mouth by the sucking process. I'll explain the milk ejection process also. This is the what is internal structure. So sometime in our examination asking identify the chronological order of movement of milk from mammary gland to into the baby mouth. Okay, so milk production started from where? Alveoli, and finally these are released out through which duct? Lactiferous duct. So this is the order. Okay, so now and in each mammary gland how many mammary lobules are there? 15 to 20 mammary lobules are there. It is very clear, and it is. Now I'll write the chronological order. I'll write the chronological order of milk movement. Okay, first what is here? Alveoli. Okay, the group of alveoli are open into the mammary ductules. Mammary ductules. Okay, the group of mammary ductules are fused and form the mammary duct. Okay, mammary duct is open into the what is mammary ampulla. Mammary ampulla it is. Okay, the mammary ampulla it is open into the lactiferous duct. It is open into the lactiferous duct. The lactiferous duct it is open out through the nipple. It is open into the nipples or nipple. The nipple is okay, through the nipple the milk is eject into the baby mouth. Milk eject okay into baby mouth. Into baby mouth. Milk eject into baby mouth. Okay, so this is the what is chronological order of movement of milk from alveoli to mammary ductules, mammary ductules to mammary duct, mammary duct to mammary ampulla, mammary ampulla to lactiferous duct, lactiferous duct to nipples, nipples to baby mouth by the sucking. Okay, so this is the chronological order of what is milk movement, milk transportation from mammary gland to into the next accessory duct. Okay, here some important points I'll explain to you. Again, okay, after when the milk production is started, so up to here we are discussed డెవలప్మెంట్ ఆఫ్ మమ్మరీ గ్లాండ్. ఓకే, ఇప్పటి వరకు మనం ఏం చెప్పుకున్నామంటే మమ్మరీ గ్లాండ్ ఎలా డెవలప్ అవుతుందో చెప్పుకున్నాం అండ్ ఏ హార్మోన్స్ యూజ్ అవుతాయో చెప్పుకున్నాం. ఇప్పుడు మిల్క్ ప్రొడక్షన్ ఎప్పుడు స్టార్ట్ అవుతుంది అనేది ఇంపార్టెంట్. ఓకే, మిల్క్ ప్రొడక్షన్ బిగిన్స్ ఆఫ్టర్ పార్చురేషన్. ఇన్ సమ్ పర్సన్స్ బిఫోర్ పార్చురేషన్ ఆల్సో the milk production takes place. Understood? So this is one point. And which hormones are essential for production of milk and which hormones are essential for ejection of milk from mammary gland to outside? Okay, these 2 points are there. Again here the hypothalamus has major role. What is the role of hypothalamus? Hormonal regulation of milk production and ejection. Okay, first I'll write this point here. Hormonal regulation, hormonal regulation of milk production and milk ejection, milk production and milk ejection. Okay, so these 2 points I'll explain within the hormonal regulation. And as usually as usually here what is this? See, hypothalamus has major role. First, hypothalamus is there. Hypothalamus is there. Okay, the hypothalamus, what is, when it is receiving the signals from the baby sucking. Okay, it is receiving the signals. Okay, it is send the positive feedback signals. It is send the positive feedback signals to anterior pituitary, okay, also called as a adenohypophysis. These anterior pituitary can release 2 hormones here. One is the prolactin hormone and second one is the oxytocin. So prolactin PRL indication short form, oxytocin OT. The prolactin hormone which is act on alveoli. Okay, the alveoli are involved in the production of milk. Okay, milk production here takes place. Milk production, it is takes place here. Okay, after completion of the production of milk from the alveoli, okay, the oxytocin which is act on, what is smooth muscles in mammary duct. Smooth muscles in the wall of mammary duct. In the wall of mammary duct. That is one. And the smooth muscles are get contracted. Thereafter, okay, milk ejection takes place. Okay, milk ejection. It is milk ejection takes place. Milk ejection. So in the milk ejection process, what happened? After completion of the production of milk, the milk can stored within the mammary ducts. So how the mammary duct contain milk released from inside mammary gland to outside, outside means into the baby mouth, means the action of oxytocin on the wall of mammary duct. So the mammary ducts are internally lined by the smooth muscles. On the smooth muscles, the oxytocin, when the oxytocin hormone is act, the muscles are get contracted. After contraction of mammary duct, whatever milk is present inside the mammary duct, the milk is can push it out from inside mammary gland to outside into the baby mouth. Into the baby mouth. Very clear information. So this is the hormonal regulation for milk production, milk ejection. Simple thing. For the development of mammary gland, estrogen, progesterone hormones. For the production of milk, the prolactin act on alveoli. After completion of the milk production, the oxytocin act on the wall of the mammary ducts for sending the milk from inside mammary gland to outside. So that process is called milk ejection. After milk ejection, that milk is enter into the baby mouth. Okay, so this is the hormonal regulation of what is milk mammary gland for the development. After parturition, for the production of milk and ejection of milk is done by this. See, one more clarity here. ఇక్కడ ఇంపార్టెంట్ క్లారిటీ ఉంది ఒకటి. మిల్క్ ప్రొడక్షన్ ఒక వర్డ్ డిఫరెంట్, మిల్క్ ఎజెక్షన్ వర్డ్ డిఫరెంట్. ఓకే, మిల్క్ ప్రొడక్షన్ అన్న మిల్క్ సెక్రేషన్ అన్న ఒక్కటే మీనింగ్ వస్తుంది. ఓకే, విచ్ హార్మోన్ ఈజ్ ఎసెన్షియల్ ఫర్ సెక్రేషన్ ఆఫ్ మిల్క్ అంటే ప్రొలాక్టిన్ వస్తుంది. విచ్ హార్మోన్ ఈజ్ హెల్ప్స్ టు రిలీజింగ్ ద మిల్క్ ఫ్రమ్ మమ్మరీ గ్లాండ్ మీన్స్ ఆక్సిటోసిన్. అంటే మిల్క్ ఎజెక్షన్ అన్న మిల్క్ రిలీజింగ్ అన్న ఒక్కటే మీనింగ్. అది మిల్క్ ఎజెక్షన్ మీన్స్ ప్రాసెస్ ఆఫ్ రిలీజింగ్ ఆఫ్ మిల్క్ ఫ్రమ్ మమ్మరీ గ్లాండ్ టు ఇంటూ ద బేబీ మౌత్. ఓకే, మిల్క్ ప్రొడక్షన్, మిల్క్ సెక్రేషన్ ఈజ్ సేమ్, మిల్క్ ఎజెక్షన్, మిల్క్ రిలీజింగ్ ఈజ్ సేమ్ మీనింగ్ పాయింట్స్. ఓకే, సో యూ షుడ్ కీప్ ఇన్ యువర్ మైండ్ దీస్ టూ వర్డ్స్ డిఫరెన్స్. సో మీరు 100% ఈ డిఫరెన్స్ని ఫైండ్ అవుట్ చేస్తే ఖచ్చితంగా మీకు ఇందులో నుంచి ఏ బిట్స్ వచ్చినా నీట్ ఎగ్జామ్లో ఆబ్జెక్టివ్ పాయింట్లో 100% మనం సాల్వ్ చేసుకోగలుగుతాం. వెరీ ఇంపార్టెంట్ ఇది. సో ఈ వర్డ్స్ని మనం డిఫరెన్షియేట్ చేయాలి. సో ఇది మనకు ఒక క్లారిటీ. దిస్ ఈస్ ద అప్ టు హియర్ ఇట్ ఈస్ ద ఫిమేల్ రిప్రొడక్టివ్ గ్లాండ్స్ ఇన్ఫర్మేషన్. ఇట్ ఈస్ స్కెన్స్ గ్లాండ్స్, Bartholin glands and mammary glands information, including hormonal regulation also. We are clearly discussed this point. Okay, after completion of this, next we'll discuss the oogenesis. Okay, next we will discuss the oogenesis process. Okay, it is next gametogenesis. In the next gametogenesis process, gametogenesis. In the next gametogenesis process, what is that is the production of female gamete. Production of female gamete. Production of female gamete. Okay, it is the ovum. Okay, production of ovum is called oogenesis. Production of female gamete is called oogenesis. Production of female gamete is called oogenesis. Okay, now we will discuss the oogenesis process here. Now we will discuss this. Okay, in the oogenesis process, okay, before this, before the phases here, in previous classes we are discussed the spermatogenesis. Okay, the spermatogenesis begins in human male at the puberty age only, but in human females the oogenesis it is begins at when the female is present in fetal age. In this time only what is the oogenesis begin. Very simple, oogenesis begins at fetal stage. Okay, oogenesis begins at female fetal stage or fetal stage of the female fetal stage or fetal age or fetal stage it is. So generally for the oogenesis the gonadotrophins are essential, okay, including in spermatogenesis are also the gonadotrophins are essential. Generally the own gonadotrophins are produced at the puberty age only. The own gonadotrophins are produced at the puberty age. But when the female is present in fetal stage, how can the own gonadotrophins are produced by that female fetus anterior pituitary? It is not possible. Then without the own gonadotrophins, how can the ovaries of this female fetus can stimulate and initiate the oogenesis? ఓకే, ఇప్పుడు జనరల్గా oogenesis బిగిన్ కావాలి, స్టార్ట్ కావాలి అంటే గోనాడోట్రోఫిన్స్ అవసరం. ఇప్పుడు మేల్ రిప్రొడక్టివ్ సిస్టంలో స్పెర్మాటోజెనిసిస్ మనం డిస్కస్ చేసినాం. ఇలా డిస్కస్ చేసేటప్పుడు స్పెర్మాటోజెనిసిస్ స్టార్ట్ అయ్యేది గోనాడోట్రోఫిన్స్ తోటి, ఎఫ్ఎస్హెచ్ ఎల్హెచ్ తోటి. అప్పుడు 12 to 14 ఇయర్స్ ఏజ్ వచ్చిన తర్వాతనే స్పెర్మాటోజెనిసిస్ స్టార్ట్ అవుతుంది హ్యూమన్ మేల్లో. ఎందుకంటే ఆ ఏజ్లోనే పిట్యూటరీ గ్లాండ్ హైపోథాలమస్ నుంచి గోనాడోట్రోఫిన్ రిలీజింగ్ హార్మోన్ వస్తుంది. ఆ గోనాడోట్రోఫిన్ రిలీజింగ్ హార్మోన్ యాంటీరియర్ పిట్యూటరీ స్టిమ్యులేట్ చేస్తే యాంటీరియర్ పిట్యూటరీ అనేది ఎఫ్ఎస్హెచ్ ఎల్హెచ్ హార్మోన్ని ప్రొడ్యూస్ చేస్తుంది. ఈ ఎఫ్ఎస్హెచ్ ఎల్హెచ్ హార్మోన్స్ అనేవి స్పెర్మాటోజెనిసిస్ని ఇనిషియేట్ చేసి స్పెర్మ్ ప్రొడక్షన్లో ఇన్వాల్వ్ అవుతాయి హ్యూమన్ మేల్లో, అది ప్యూబర్టీ ఏజ్లో. ఓకే, బట్ ఇక్కడ ఫిమేల్ హ్యూమన్ ఫిమేల్లో oogenesis అనేది క్వైట్ ఆపోజిట్ ఉంటుంది. ఎట్లా అంటే oogenesis బిగిన్ కావడానికి ఓన్ గోనాడోట్రోఫిన్స్ అవసరం లేదు. నో నీడ్ టు దేర్ ఓన్ గోనాడోట్రోఫిన్స్ ఫర్ ఇనిషియేషన్ ఆఫ్ oogenesis. Then how the gonadotrophins are how the gonadotrophins are available for initiation? సో ఇంకొకటి, అసలు గోనాడోట్రోఫిన్స్ లేకపోతే oogenesis బిగిన్ కాదు. ఇప్పుడు ఓన్ గోనాడోట్రోఫిన్స్ లేవు, బట్ oogenesis స్టార్ట్ అయింది. ఎలా? దెన్ హౌ? మీన్స్ మదర్ గోనాడోట్రోఫిన్స్. జనరల్గా ఎంబ్రియానిక్ డెవలప్మెంట్లో ఆఫ్టర్ కంప్లీషన్ ఆఫ్ ద థర్డ్ మంత్ ఆఫ్ ద జెస్టేషన్ పీరియడ్, ఓకే, థర్డ్ మంత్ జెస్టేషన్ పీరియడ్లో ఏమవుతుందంటే కంప్లీట్ ఆర్గానోజెనిసిస్ పూర్తవుద్ది. అంటే ఇంటర్నల్ ఆర్గాన్స్ మొత్తం ఆర్గానోజెనిసిస్ కంప్లీట్ అయిపోద్ది. అంటే ఓవరీస్ అనేవి వన్ ఆఫ్ ద ఆర్గాన్ కదా, దెన్ దీస్ ఓవరీస్ డెవలప్మెంట్ ఆల్సో కంప్లీటెడ్. See, into the fetal blood, how the mother gonadotrophins are enter into the fetal blood or fetus? Means these hormones are crossed through the placenta along with the nutrients. Very important thing. Okay, along with the nutrients, these gonadotrophins are transferred into the fetus through the placenta. Once the mother gonadotrophins like FSH, LH, which are enter into the fetus blood, and once these are enter into the fetal blood, what is the nature of these gonadotrophins? Stimulation of gametogenesis, either in male or in female. But in male, the gametogenesis begins with own gonadotrophins. But in female, the oogenesis begins with mother gonadotrophins. ఎందుకంటే ఫీటల్ స్టేజ్లో ఉన్నప్పుడు ఆ ఫీటస్ యాంటీరియర్ పిట్యూటరీ అనేది గోనాడోట్రోఫిన్స్ ప్రొడ్యూస్ చేయలేదు. ఆ గోనాడోట్రోఫిన్స్ ప్రొడ్యూస్ కావాలి అంటే ఆ ఫిమేల్ అనేది పార్చులేషన్ తర్వాత ప్యూబర్టీ ఏజ్కి రావాలి, ఓన్ గోనాడోట్రోఫిన్స్ రావాలంటే. బట్ ఇక్కడ ప్యూబర్టీ ఏజ్లోనే ఫీటల్ ఏజ్లోనే స్టార్ట్ అవుతుంది oogenesis. ఓకే, సి ద oogenesis what is begins at female fetal stage only with the mother gonadotrophins. Fetal oogenesis with mother gonadotrophins. Mother gonadotrophins it is. With mother gonadotrophins it is one. And how the mother gonadotrophins are enter into the fetus body means through the placenta as usually. ప్లాజెంటా నుంచి క్రాస్ అవుతాయి. సో ఇది ఫస్ట్ oogenesisలో మనం డిస్కస్ చేయాల్సిన ప్రాసెస్. ఫీటల్ స్టేజ్లో స్టార్ట్ అవుతుంది ఇక్కడ. Okay, female fetal stage లో స్టార్ట్ అవుతుంది oogenesis. Okay, it is first point. Within the oogenesis, 3 phases are there in the oogenesis process. 3 phases. Okay, in the spermatogenesis process 4 phases are there. Okay, just remember that what are the 4 phases of spermatogenesis. One is the multiplication, second one is the growth phase, third one is the maturation, and fourth one is the differentiation. Okay, and in oogenesis 3 phases are there. In these 3 phases, one is the multiplication. Okay, same as in spermatogenesis first phase, here also first phase it is the multiplication, it is first one. Okay, and second one is the growth phase, same as in second spermatogenesis, it is also same growth phase, multiplication phase and growth phase, these two are there. Okay, and next third phase is the maturation phase. Third phase is the maturation phase, it is third phase is the maturation phase. Okay, third phase is the maturation phase, and there is no differentiation phase in oogenesis like spermatogenesis. Okay, so these 3 phases are there, same as in spermatogenesis. In a multiplication phase, in the multiplication phase, what are mitotic divisions, repeated mitotic divisions are takes place here. Repeated mitotic divisions are takes place, it is one point. Next what is number of oogonal cells are increased, number of oogonial cells, number of oogonial cells, how many means approximately couple of million oogonial cells are produced, couple of million oogonial cells are produced here. So this is, and by the repeated mitotic division, same as spermatogenesis, by the repeated mitotic divisions that oogonal cells and as well as in spermatogenesis, spermatogonal cell size is minimized. For increasing the size of the cell, these all cells are enter into the growth phase. In this growth phase, what happened? By the increase the concentration, okay, increase the size of the cell, increase the size, okay, increase the size, increase the size of the cell by the increasing the concentration of chromatin, same as spermatogenesis here also, by the increasing by the increasing the size of the chromatin increasing the size of the chromatin, increasing the size of the chromatin, this is by the increasing the size of the chromatin, these are. So after completion of the growth phase, all our primary oocytes are developed. The primary oocytes are enter into the what is maturation phase. The maturation, it is begins at where it is begins at, it is maturation, it is started or begins from puberty age, begins from puberty, begins from puberty age, it is begins from puberty age. Okay, it is begins from puberty age, and here only we are observe the meiosis divisions, we are observed here meiosis divisions here, meiosis divisions, we are observed here that meiosis divisions, it is very clear. This is the what is oogenesis, how many phases are there, in which phase what happened, we are clearly discussed. Now we'll discuss with the help of one particular chart, that is the oogenesis process. Okay, come to the point here. First here what oogonal mother cells. So first phase it is multiplication, multiplication it is. Okay, it is the first one, multiplication. Okay, so the multiplication it is start from the oogonial mother cells at the fetal stage. It is oogonial mother cells. So where the oogonial mother cells are present? Oogonial mother cells are present outer lining of the ovary. Outer layer of the ovary is what? Sperma. Oogonial cells, oogonial layer, epithelial cells are there. The epithelial cells nothing but oogonial mother cells. The oogonial mother cells are initiate the oogenesis process. After that, these oogonial cells are okay enter continues to the repeated mitotic divisions. Continues to the repeated mitotic divisions. By the repeated mitotic divisions, what are oogonial cells are formed here. These are the daughter cells. Okay, these are the oogonial cells are formed. See, the oogonial cells are also again which are involved in the repeated mitotic divisions here. Again, these are involved in the repeated mitotic divisions. By the repeated mitotic divisions, okay, again more number of here again that mitotic divisions. So many times, not only 2 or 3 times, many times the mitotic divisions are what is continue. So again oogonial cells are formed here. See, these oogonial mother cells, oogonial cells, these are all are present in diploid state, which means what is present in 46 number of chromosome condition. These are all are present in oogenesis, what is diploid state, and these are all are present in 46. Diploid state means 46. Up to here, from here to here, it is the multiplication. Up to development of oogonial cells, how many? Couple of oogonial cells are formed here. Couple of oogonial cells are formed. Okay, these all cells are present in diploid, right? After that, these all cells are by the repeated cells, the small sized cells are formed here. Small size is decreased, minimized. By the repeated divisions, what happened? These all cells are enter into the growth phase. These are all are enter into the growth phase. These are all are enter into the growth. Means, already I clearly explained. Growth means increase the size of the cell by the increasing the size of the chromatin inside the nucleus. Okay, so very important thing. So what happened in this? These all oogonial cells are completes their what is growth phase, and the size of the cell is increased. So after completion of the growth phase, the increased oogonial cell is called the primary oocyte. It is called a primary oocyte. Very important thing, it is primary oocyte. Now the primary oocyte also present in diploid and 46 number of chromosomes, same as primary spermatocyte. Okay, same as primary spermatocyte. The primary oocyte is also present in diploid state with 46 number of chromosomes. Understood? So this is, and so up to the formation of primary oocyte, these are present in the fetus up to which time the fetus is present inside the mother uterus. These are all are present up to the mother uterus. These are present. See, within the primary oocyte, the further divisions are will be stopped. Means temporarily, as arrested, like that. Further divisions are arrested temporarily. Further divisions are suspended. Further divisions are suspended. Means after some time the divisions are will begin as usually. After some time the divisions are will begin as usually. Understood? Means the primary oocytes, okay, what is meiosis 1, these are arrested, and what is metaphase 1, meiosis 1, metaphase 1, these are temporarily arrested, temporarily arrested, temporary arrested, temporarily arrested these are. So when the meiosis 1, metaphase 1, these are completed in the primary oocyte, means when the female comes to the puberty age with the own gonadotropins, the primary oocytes are completes the meiosis 1 and metaphase 1 also. Very important and interesting point is there here. When the primary oocyte completes the meiosis 1 and metaphase 1, means when that female is comes to maturity age, puberty age, okay, at the puberty age the hypothalamus can release gonadotropin releasing hormones. The gonadotropin releasing hormones are reached to the anterior pituitary. The anterior pituitary can release FSH, LH, and these FSH and LH hormones are initiate the these primary oocyte divisions. So after completion of the meiosis 1 and metaphase 1, okay, the secondary oocyte will be developed. Okay, in this, how many number of oogonal cells are enter into the growth phase? The same number of primary oocytes are formed. For example, couple of millions of primary oogonal cells are complete the growth phase. Then how many number of primary oocytes are produced? Same the couple of number. Same the couple of number of primary primary oocytes are formed. Understood? Same reason, in the growth phase there is no mitotic, there is no meiotic. That's why there is no chance for increasing and decreasing the number of what is these oogonal cells as well as primary oocytes. Okay, for example, 1 lakh of oogonal cells are to complete the growth phase, 1 lakh of primary oocytes are produced. Okay, up to which age? Okay, up to which age the primary oocyte production takes place? That is the question. Okay, up to the parturition, the new primary oocytes are produced. Understood? Up to the parturition, the new primary oocytes are produced. Once the delivery completed, once the baby comes out from mother uterus to outside, immediately the primary oocyte number will not increase, and finally the oocyte number gradually decreased by the degeneration of primary oocytes. By the degeneration of primary oocytes. Okay, so this is the what is oocyte, and okay, in this stage, okay, in the primary oocyte stage, what is the fetus delivery? Fetus delivery, nothing but it is a better word, after delivery. Okay, after delivery, after delivery of baby, after delivery of baby, okay, what is these are all are arrested, and when the baby comes to puberty age, when the female comes to maturity age, when the female is comes to puberty age, puberty age, puberty age, okay, within primary oocyte, primary oocyte, primary oocyte completes, primary oocyte completes meiosis 1 and metaphase 1. Meiosis 1 and metaphase 1, these both are completed. Okay, and one more important point is there here. at the time of parturition, I'll take one more slide here. At the time of parturition, very important point. Okay, at the time of parturition, at the time of parturition, okay, parturition, primary oocytes are couple of million are there, couple of million, and when female comes to puberty age, female reach to puberty age, only 60 to 80,000 primary oocytes are there. 1000 primary oocytes are left. Primary oocytes are left in each ovary. Left in each ovary. It is very, very important word. Means from parturition to, after parturition already I told na, బేబీ డెలివరీ తర్వాత ప్రైమరీ oocyte నెంబరు అంటే న్యూ ప్రైమరీ oocytes ఉండవు, అంటే పెరగవు, న్యూ ప్రైమరీ oocyte ప్రొడక్షన్ జరగదు. ఫస్ట్ థింగ్, న్యూ oocyte పెరగకపోగా ఆల్రెడీ ఏవైతే ప్రైమరీ oocytes ఉన్నాయో పార్చురేషన్ నుంచి ప్యూబర్టీ ఏజ్ వచ్చేసరికి, పార్చురేషన్ టైం అప్పుడు ఈచ్ ఓవరీలో కపుల్ ఆఫ్ మిలియన్ ఉన్నాయి. సో ప్యూబర్టీ ఏజ్కి వచ్చేటప్పటికి, ఓకే, ప్యూబర్టీ ఏజ్ అంటే 12 to 14 years, అంటే పుట్టిన దగ్గర నుంచి ప్యూబర్టీ ఏజ్, ఈ 12 years గ్యాప్లో ఏమైంది, కపుల్ ఆఫ్ మిలియన్స్లో ఉండే ప్రైమరీ oocytes ఈచ్ ఓవరీలో 60 to 80,000కే వచ్చేసినాయి. అంటే ప్రైమరీ oocytes ఏమైనా ఇక్కడ డీజనరేట్ అయిపోయినాయి. ప్రైమరీ oocytes అనేవి డీజనరేట్ అవుతూ వచ్చేస్తున్నాయి. ఓకే, సో ఐల్ ఎక్స్ప్లెయిన్ ద రీజన్. వాట్ ఈస్ ద రీజన్ ఫర్ stoppage of menstrual cycle at a particular age of the female? So what is the reason, the cessation of menstrual cycle in a particular age of the female? One question is there. That reason is the absence of primary oocytes within the ovary. That is the major reason for cessation of menstrual cycle. Okay, I'll explain. After this, I'll explain the menstrual cycle. That is, so in each ovary 60 to 80 primary oocytes are left in each ovary. That's enough at the puberty age. Okay, and what is the reason means the degeneration of primary oocytes takes place. Degeneration of primary oocytes takes place. That's why. Okay, so okay, when that female is reached to puberty age, okay, it is the primary oocyte, and at the puberty age, what is the presence of gonadotropin releasing hormone and gonadotropins means what, FSH and LH hormones. Okay, the meiosis 1 and metaphase 1 which are complete. After completion of the meiosis 1 and metaphase 1, this meiosis is the ameiosis process. It is the ameiosis, means unequal sizes daughter cells are formed. Unequal sized daughter cells are formed here. Unequal sized daughter cells. See, one is small in size and second one is large in size. Okay, the small one is the first polar body. And second one is the secondary oocyte. Large one is the secondary oocyte. Now it is present in diploid. The primary oocyte is present in diploid. It is clear. And the secondary oocyte is present in haploid in nature, means 23 number of chromosomes are there after meiosis 1. Okay, again within the secondary oocyte, what is meiosis okay, after this meiosis 2 and prophase 2, meiosis 2 and prophase, meiosis 2 and prophase 2, these two are meiosis 2 and prophase 2, again these are temporarily arrested, again these are the temporarily arrested. Okay, temporarily arrested, it is one. When the meiosis 1 and prophase 1 are what is completed in secondary oocyte, means after entry of what is sperm. Okay, after entry, very important point, after entry of sperm, after entry of sperm into secondary oocyte, after entry of sperm into secondary oocyte. Okay, what is meiosis 2 and prophase 2 will completed, will be complete, that's enough, will be complete, that's enough, after entry of sperm only. Okay, into to this woman, if the sperm is available to this secondary oocyte, sperm is available, for example it is the sperm. Okay, it is the sperm. It is the sperm. Sperm is entered. After entry of sperm into the secondary oocyte, what happened in this? Okay, so it is also what is a meiosis. It is also a meiosis process. It is also a meiosis. Okay, in this two daughter cells are formed. One is the small one and second one is the large one. The small one is called as a second polar body. It is called as a second polar. It is second polar body, and large one is called mature ovum, and it is also called as a ooted. It is also called as a ooted. Now this ooted also present in haploid 23 number of chromosomes. Now this nucleus of the what is ovum and the nucleus of the sperm both are fused and form the zygote. After that the zygote is formed. After that the zygote. After that the zygote is formed. The zygote is present in diploid and 46 number of chromosomes are there. Okay, it is the what is complete oogenesis process. See, the spermatogenesis is the 1:4 ratio. 1:4 ratio is the spermatogenesis, and the oogenesis is the 1:1 ratio. It is oogenesis is the 1:1 ratio. Oogenesis, simple thing. From single primary spermatocyte, how many number of spermatozoas are formed means 4. Okay, 2 X chromosome contains sperm, 2 Y chromosome contains sperm. And from one primary oocyte, how many secondary oocyte or how many ova are produced? How many ovum produced? Means only one. See, after completion of the meiosis 1, the mature secondary oocyte single one is produced, and first polar body it is degenerate after some time. It is not able to fertilize with sperm. The polar bodies are not able to fertilize with sperm. Okay. That's why it is degenerate after some time. Next, the second, after entry of sperm into the secondary oocyte, it is complete the meiosis again, meiosis 2. After that again second polar body and mature ovum is produced. The second polar body is also immature, means with the second polar body also what is the sperm is not able to fertilize it with second polar body also. Sperm is not able to fertilize it with. That's why again this mature worm is also only fertilized with sperm here. That's why here what is the second what is a zygote is formed here. After that what happened here? Zygote is formed. Now these polar bodies are degenerate. Finally only one from one primary oocyte only one worm is produced. That's why it is the 1:1 ratio, and here 1:4 ratio. And some questions are there, and for example it is present in diploid state, means 46 number of chromosomes, 2 X chromosomes and 44. And here also what is 1 X chromosome plus 22 and plus 22, and here also X chromosome plus 22, X chromosome plus 22. So single ovum has 1 X chromosome. Okay, so then means in every 100 percentage of sperms, 50 percentage of sperms are having X chromosome, 50 percentage of sperms are having Y chromosome. But here the 100 percentage of ova are having X chromosome only. Only X chromosome is there here like this. So then this is the important points regarding the number of chromosomes formation. And here some questions are there based on spermatogenesis and oogenesis. Okay, for example that question is there. That question is okay. That question is, what is that? In a seminiferous tubule, in a seminiferous, in a seminiferous tubule, in a seminiferous tubule, okay, 1000 primary spermatocytes are present. 1000 primary spermatocytes. 1000 primary spermatocytes are there. Okay, 1000 primary spermatocytes are present. Okay, then how many? Okay, then how many secondary spermatocytes and what is spermatozoas? Spermatozoa are produced. Okay, here option 1 again 1000 produced respectively. You should use respectively word, respectively. Okay, first you find out the secondary spermatocytes. They can second you find out the spermatozoa, and here 2000. Okay, next option B 2000 and 4000, and option C 3000 and 6000, option D 4000 and 8000. So what is the answer? You find out the answer and send again reply to me in your videos. You send message to me. Then how many number of primary spermatocytes are produced? How many number of primary spermatozoas are produced by the from the 1000 primary spermatocytes? Okay, so I want this answer. Then who will send firstly? Then I'll see. And the next question is there. Okay, regarding the female reproductive system. So within the nowadays where within the ovum, within the ovary, okay, with in the ovary, okay, within the ovary, okay, how many, again same, I'll give the same number, 1000 primary oocytes are there, primary oocytes, 1000 primary oocytes are there, 1000 primary oocytes are there, oocytes present, 1000 primary oocyte present, okay. Then how many, how many secondary oocytes and ootids are ovum, ootids are ova, simply it is, are produced respectively, produced respectively, it is one. Then options here, A option 1, 1000 and 1000, same options here, then I'll change here, and 2000, 2000 or 1000 and 2000, option C 1000 and 3000, option D 1000 and 4000, 1000 and 4000. Then what is answer for question 1, first question, and what is answer to second question? As my class, you tell and you write it, this answer within the link, okay. Some message option is also there, you can send the answers to me, okay, very important. And based on this, so many questions are there here, so many questions, okay. For example, one more question is there, some questions I'll frame some questions here, and question number 3, third question is there in this, in a what is in a ejaculation, in a ejaculated semen, ejaculated semen, in ejaculated semen, okay, what is 1 lakh of spermatozoas are present, 1 lakh of spermatozoa, 1 lakh of spermatozoa are present, okay, which are produced from, which are produced by, which are produced by how many, how many primary oocyte, primary spermatocytes, how many primary spermatocytes, very important. So I want answer directly, I don't give any options, so I want answer, okay. You write down the answer in message, okay. So very important point, so many questions are there based on this, reversely, from primary spermatocytes to spermatozoa, spermatozoa to primary, just reverse, we are reverse that, okay. So this is one important point, right, these are the what is oogenesis process, okay. After that we will discuss in the next okay. So these are the some questions based on what is this previous class and this class. Next we will go to follicular development, okay. Now we will discuss the follicular development, okay. It is follicular development, follicular development, okay. So the follicular development process, it is begins from the puberty age only. Okay, follicular development, it is start from puberty age. Start from puberty age. Okay, start from puberty age means from 12 to 14 years age. 12 to 14 years age. From 12 to 14 years age, what is the follicular development process started? See, once again I will draw the ovary. See, this is the ovary. For easy understanding, again I will I am drawing this ovary picture. So, it is the ovary. And where does the various stages of follicles are found? Means within the stromal cortex area. Within the stromal cortex, various stages of follicles are found. Various stages. Various stages of follicles are found here. It is. Various stages of follicles are found in stromal cortex. This total area is the stromal cortex. Stromal cortex. Okay, in a previous class we have discussed this. Okay, right. Here the primary follicles are found. These are the primary oocyte. Okay, so once the primary oocyte is developed by the completion of growth phase, these are all are present as these are all are get surrounded by one layer of follicular cells, one layer of granular cells simply. The granular cells are called here follicular cells. Very important thing. See, it is the primary oocyte. So always I use the single color for the oocyte, that is primary oocyte. It is the primary oocyte, and surrounding the primary oocyte, what are primary follicles? For the primary follicles, I will use this red color. I will use the red color for follicular cells. See, now this is the primary follicle. It is the primary follicle. It is very important point. See, surrounding the primary oocyte, if one layer of follicular cells are present, that is the primary follicle. Okay, so these are the follicular cells. These are the follicular. These are the follicular cells. These are the follicular cells. It is one. Okay. See, at the puberty age, what are two gonadotrophins? One is the FSH and the second one is the LH. Okay, FSH and LH, these are there. These FSH and LH hormones are, okay. Now these FSH hormone stimulating to the primary follicle. Very simple and interesting point it is. The follicular stimulating hormone stimulating to the primary follicle. Now the primary follicle, it will gradually increase their size. Okay, when the primary, what is FSH stimulating to the primary follicle, again the number of follicular cells are increased. Still it is the primary oocyte, and surrounding the primary oocyte, what is the follicular cell number will increase. Follicular cell number is increased. Means here, how many rows of follicular cells are developed here? 2 rows of follicular cells are developed. Okay, these 2 rows of follicular cells contain the structure is the secondary follicle. It is the secondary follicle. Very simple and interesting thing is there. If surrounding the primary oocyte single layer of follicular cells are there, that is the primary follicle. If surrounding the primary oocyte two rows of follicular cells are present, that is the secondary follicle. That is the secondary follicle. Still the FSH hormone is act on. Still the FSH hormone act on the secondary follicle. Now which is again developed into the tertiary follicle. Again it is the actually it is the primary oocyte. Initially it is the primary oocyte. Initially it is the primary oocyte, and surrounding the primary oocyte, granulosa cells are developed with little space surrounding the two or more rows of follicular cells are there here. Here it is the two or more rows of follicular cells. Two or more rows follicular cells are there. After that, this stage is called as a tertiary follicle. Tertiary follicle. Okay, important point is there. Within the tertiary follicle, okay, what are the granulosa cells are? What is these granulosa cells are? Modified into the thecal cells. The granulosa cells are modified into the thecal cells. Okay, these thecal cells are two types of thecal cells are there. Okay, in this thecal cells are theca externa. Okay, one is the thecal cell, theca externa is one. It is the theca externa, it is one, and theca interna. Theca externa and theca interna, two layers are there. These theca externa, theca interna, which are involved in the production of estrogen. Very important point. Theca interna cells are involved in the production of estrogen. The estrogen can stimulating to the primary oocyte for the completion of meiosis 1 in primary oocyte. Now it will develop into the secondary oocyte. Now this is developed into the secondary oocyte. Now this is the secondary oocyte. It is the secondary oocyte. Okay, right, it is the secondary oocyte. Now within the tertiary, and one more important changes are there here, that is what is surrounding of this, where is this color? Okay, okay, surrounding of this, what is the antrum is formed here, the empty follicle antrum. Empty follicle is there surrounding the secondary oocyte. The empty space is formed. The empty space is called antrum. So what is the difference between secondary follicle and tertiary follicle? In secondary follicle, primary oocyte is there. In tertiary follicle, secondary oocyte. Along with the secondary oocyte, surrounding the secondary oocyte, little space is present. That space is called here antrum. Okay, that space is called here what? Antrum is there. Okay, after that, this secondary follicle, tertiary follicle, again it will develop into what is mature follicle or Graafian follicle. It is developed into mature follicle or Graafian follicle. Okay, see, this is the as usually secondary oocyte. It is the secondary oocyte. Okay, and as usually these are the granulosa cells. These are the granulosa cells. Okay, or follicular cells. These are arranged developed in more than two rows. Okay, and size is also very big. Along with this, okay, surrounding the it is the secondary oocyte. It is the secondary oocyte. Okay, surrounding the secondary oocyte, one layer is secreted by the secondary oocyte lemma. That lemma can synthesize one layer. That layer is called zona pellucida. That is the zona pellucida, and this is the non-cellular layer. It is the non-cellular membrane. It is the non-cellular membrane. Okay, ఇది సెల్స్ లేకుండా ఫామ్ అవుద్ది, సెక్రిషన్స్ తోటి ఫామ్ అవుద్ది అన్నట్టు. ఇది అంటే ఊలెమ్మ కొన్ని సెక్రిషన్స్ సెక్రెట్ చేస్తుంది. ఆ సెక్రిషన్స్ రిజిడ్ అయి ఒక లేయర్ని ఫామ్ చేస్తుంది. ఆ లేయర్ని మనం జోనా పెల్యూసిడా అంటాం. ఇట్ ఈస్ ఫస్ట్ లేయర్. ఓకే, it is more close to the what is secondary oocyte. After that, some granular cells are differentiate from the inner lining of granular cells, and which are arranged into the one more layer, and surface of the what is the zona pellucida. This layer is the corona radiata. That is the corona radiata layer. Okay, these two, and the corona radiata is the cellular. It is the cellular layer. Corona radiata is the cellular layer. It is one. Along with this, some granulosa cells are what is in this process. The total secondary oocyte and surrounding layer, what is, it is move a peripheral side. In the antrum, it is move a peripheral side. It is move a peripheral side. So when it is move a peripheral side, the granular cells are attached to the secondary oocyte. So these modified granular cells are also called as a cumulus oophorus. These are also called as a cumulus oophorus. Cumulus oophorus layer. So this cumulus oophorus layer, what is attaching these what is along with these layers, the second oocyte, and the second oocyte cannot move anywhere in this antrum. See, as usually here antrum is there. Now in this along with the secondary oocyte, the ovular membranes are there surrounding the secondary oocyte. After that, it is converted into the mature follicle. The mature follicle also called as a Graafian follicle. Mature follicle also called as a Graafian follicle. Mature follicle also called as a Graafian follicle. It is very important thing, Graafian follicle. Now, how can you differentiate the Graafian follicle with tertiary follicle? Means in tertiary follicle surrounding the secondary oocyte there is no ovular membranes, but in Graafian follicles two ovular membranes are there. One is the non-cellular membrane zona pellucida. It is directly synthesizing from the ulemma secretions, and the second layer is the corona radiata. These are the cellular layers. Some granular cells are reached to the surface of the zona pellucida and which is developed into the one more layer. This is the corona radiata second layer. And in this two layer, which one is more close to the secondary oocyte? Means zona pellucida. Outer layer of the secondary oocyte, corona radiata. So this is the Graafian follicle and mature follicle. Now up to here, okay, up to here, the FSH hormone act on these what is follicular developments. After completion of the Graafian follicle, now LH hormone which is act on Graafian follicle for ovulation. For ovulation, the LH hormone is act on the Graafian follicle. Now, when it is reached to, right, it is ovulation. Now we will discuss. Say it is the ovular membranes. So when the luteinizing hormone is act on what is, when the luteinizing hormone act on what is this mature Graafian follicle, it is act on mature Graafian follicle. So it is gradually reached to the peripheral side, and peripheral outer layer of the ovary get ruptured. After rupturing the layer, okay, the secondary oocyte along with the two layers, it can be released from what is Graafian follicle to outside, it is released. Now this is the secondary oocyte. It is the secondary oocyte. It is the secondary oocyte. Okay. and the process of releasing of secondary oocyte from the graphene follicle is called ovulation. For the ovulation, which hormone is essential? LH surge. Okay, LH surge. It is helps to ovulation. This is one secondary oocyte. Okay, now after that the empty graphene follicle is formed. The empty graphene follicle is called here corpus luteum. Okay, it is the corpus luteum. Corpus luteum. Okay, so now this corpus luteum, okay, now it is the empty graphene follicle. It is the empty graphene follicle. Empty graphene follicle, that's enough, and which is released two hormones. One is estrogen and second one is progesterone. One is estrogen, second one is progesterone. These two hormones are produced by the corpus luteum. See, by the completion of menstrual cycle, gradually the corpus luteum, then what is the color of corpus? Actually, the corpus luteum color is changed here. Okay, corpus luteum color is changed. Okay, it is developed into the yellow color. Okay, what is the color of corpus luteum means? Yellow color. Actually, it is changed into yellow color, yellow mass of tissue inside the ovary. Yellow mass of tissue inside the ovary is the corpus luteum. When it is present in the corpus luteum condition, these two hormones are released. Okay, after by the end of the menstrual cycle, these empty graphene follicle is what is gradually degenerate. After degeneration, these graphene follicle is converted into white mass of tissue. Once the yellow color changed into white color, okay, it is converted into the corpus albicans. Corpus luteum, it is converted into corpus albicans. Okay, corpus albicans, and finally it is degenerate by the end of the menstrual cycle. Okay, degenerate by the end of the degenerate by the end of the corpus luteum, by the end of the menstrual cycle, by the end of the menstrual, by the end of the menstrual cycle, by the end of the menstrual cycle, the corpus luteum, it is converted into corpus albicans. In the corpus albicans condition, the hormones cannot synthesize, and again finally it is degenerate. Again the next menstrual cycle begin, and the new primary follicle again started the development. In every menstrual cycle, from each ovary, only one follicular development takes place for the development of graphene follicle. Okay, I'll explain the detailed information for the ovaries at which menstrual cycle, which ovary can produce the what is graphene follicle and causes to ovulation. We'll discuss in the next part 3 in female reproductive system in menstrual cycle. We are clearly discussed that. In part 3, we will discuss menstrual cycle and what is fertilization and embryo development. We will discuss. Maximum will complete the human reproductive system in part 3 session. Okay, right, until that, and you keep follow these videos, and what is share to your friends like who are neat aspirants are there, you can send this. Okay, thank you, we'll meet in part 3.